Today I'm going to talk to you about the metadata and how we can use them to integrate between the different data links processes. So a little overview first of all, the centers that generate data. So we got 6 centers that generate data. We got 3 on the East Coast and 3 on the West Coast. Harvard, Pro, GSOS, Eurolink, WebSQ, and the DCIC. And all these centers generate data. And they don't generate one type of data, they generate many types of data. So we got transcriptomic data like RNA-seq, some centers generate imaging data, some centers genomic data, some centers generate genomic process, and other centers do proteomic data. And the idea, of course, with links is to find a way to integrate all this data. So how do we do that? We have done very good annotations of the metadata. So about this of Google, it's a very nice explanation. Metadata are the data describing other data. And in links, we capture metadata for all the key reagents that are involved in links assays. So we capture metadata for cell lines, for other cells, small molecules, for shRNAs, for proteins, and whatever is left for the other reagents category. And there are many use cases. If you have good metadata, you can build stuff. For example, maybe you want to filter down to all those data sets that use the same cell line. You want to have L1000 data sets and imaging data sets that use the same cell line. Or you want to use the same find data sets that use the same cell line, but also are both from the same provider. Or maybe you don't care the same cell line, you just want them to be a specific cancer type, like you want breast cancer cell lines. So if you have good annotations and good metadata, you can do all these things. And here is another example. This is the typical links model. You have a small molecule here, and you have a system that you do are going to portray. So if you have metadata for the small molecule, you can go with annotations. You can go back to Campbell and find all the protein targets of the small molecule targets. If you know what the cell line is, what cell type is, cancer cell type is, and other stuff, you can go to PCGA and you get patient data, patient samples that work from the same cancer type, and then do your analysis. So that's why we need to capture a lot of metadata. And for example, for cell lines, we capture 40 different metadata fields. Fields like what was the name of the provider, or what was the catalog ID of the provider, or which sender uses the cell line, what was the organism of the cell line, and so on. For antibodies, we captured 29 fields. Again, what was the catalog ID of the where you bought the antibody, or what's the target of the antibody, and so on. For proteins, we have pure fields, and we capture stuff like the uniprot ID of that protein that was used in the assay, or if there were any modifications and mutations, and 21 fields like that. And in total, we have 9 metadata categories. You can see we capture the most fields about the cell lines, and the less fields about the other reagents categories that are targeted with everything else. And of course, everything is publicly available, so you can go and download the metadata with descriptions, the control vocabulary they use, and everything from the website, the linksproject.org website. And here I'm gonna just say 3 ways that we collect the metadata. So the first way and the second way are from the centers. Either we can collect them through an API from the center's database, or directly from the center's website. The third way is the new way, the cool way. It's our new Metadata Submission Tool. It's a very easy way for the centers to send us the metadata. So it's only 2 steps. First step, you just choose your category of the metadata. Maybe you want to send primary cell metadata. So you upload your Excel file, and then if you haven't used standardized metadata fields and use other fields, you just have a drop-down menu and you align your headers with the official metadata headers. And in 2 steps, you're done. And this is a small overview of what we do with the metadata. So it's only a 3-step overview. First, we have to obtain the metadata through the ways we talked before from the centers. And then the metadata come to the DCIC and we have to validate them. We contact vendors, or we go back to the generation centers, or we check online databases to verify that what was sent was what was meant to be sent. Then we upload them to our database, our metadata registry, and that's where we store all our metadata. And then we can create packages out of these datasets. That these datasets have both metadata and they have the real data also. So it's very easy for a user to just open the folder and see what reagents were used and the results of the experiment. And I think this is my last slide. So with this we can do a lot of stuff. Once we have the packages that everything uses the same control vocabulary and everything's aligned, you can integrate between packages from different assays, but you can also integrate between separate databases. Because the vocabulary is controlled, it's very easy, for example, to connect to the CCLE or Dental or the CTA database, or whatever reason your analysis wants to go for.